Hi, I'm Gordeev, a PhD student at the Technical University of Moldova, and I'm grateful for the opportunity to present the project I'm currently working on under the guidance of Sergey Manguz's lab at the University of Southern California, which is entitled Rigorous Benchmarking of Methods for SARS-CoV-2 Lineage Abundance Estimation in Wastewater. Wastewater genomic surveillance of SARS-CoV-2 using next-generation sequencing has many important benefits. In particular, it is scalable to communities of any size within the limits of the sewerhead population. It is cost-effective since wastewater samples contain viruses derived from multiple individuals. It is non-invasive, meaning that no interaction with sampled individuals is required. Compared to clinical genomic surveillance, it provides more balanced estimates of variant prevalence in communities, covering underserved, vulnerable, unsampled, and asymptomatic individuals. Importantly, it enables the detection of emerging variants but also surges in variant transmission several weeks earlier than reflected in the clinical data. And also, compared to PCR-based SARS-CoV-2 surveillance, it offers full genomic information, enabling detection of emerging and novel variants, including cryptic ones. These benefits make it well suited for passive surveillance in the post-pandemic era, and its relevance is even higher now given that the rate of clinical testing and sequencing of SARS-CoV-2 have plummeted. To estimate the prevalence rates of SARS-CoV-2 viral lineages in the population, the relative abundances of the lineages have to be inferred from the wastewater sequencing data. Since SARS-CoV-2 RNA has to be sequenced from a dominant genomic background of our species, targeted sequencing using specially designed amplicon panels is typically performed. The obtained reads are used by bioinformatics methods that are normally able to both identify the lineages present in the sample and estimate the relative abundances. Certain challenges apply here. In particular, the fact that the samples represent mixtures of several highly similar genomes derived from multiple individuals. Also, due to the reliance on amplicon sequencing, low-quality samples may suffer from variable amplicon abundance, including amplicon dropout. So essentially, different pieces of the same genome can have different frequencies, and certain pieces might be missing from the data altogether. Our challenges apply as well. Numerous bioinformatics methods have been developed or repurposed for wastewater surveillance. However, there are very few studies comparing these methods systematically, and these studies have important limitations, such as the fact that they use only simulated data or cover just a handful of tools. In our study, we plan to benchmark all methods that were used or considered for resolving the lineage composition of wastewater samples. This includes methods that were specifically developed for wastewater surveillance, colored black in the table, as well as repurposed methods. For instance, repurposing the RNA-seq method, indicated here with blue color, is very straightforward. In place of the transcript sequences used as input, we just provide the lineage genomes. According to the general algorithmic approach, these computational methods fall in two main categories. Methods using a read classification approach, underlined in this table, assign each read either deterministically or probabilistically to individual reference genomes, according to the signature mutations they share. Next, they aggregate the read counts by lineage to estimate their total abundance. Maximum likelihood estimation via expectation maximization is normally used in this case. The second big group involves methods using a deconvolution approach, marked with bold here. These are applied to a list of mutation frequencies obtained by aligning the reads to a reference and use the least squares method to quantify lineages. For our benchmark, we use both real and simulated datasets. To simulate our datasets using realistic lineage mixtures that would mirror the prevalence ratios of viral lineage witnessed during the pandemic, we extracted weekly count data for co-occurring SARS-CoV-2 lineages as reported on the GISAID database for the United States between the January 2020 and December 2022. Only lineages with a weekly case count greater than 10 were considered. Similarly, we extracted the percentage values of co-occurring Delta and Omicron sublineages and their parent lineages as reported every second week between November 2021 and March 2023 on the NextTrain database. 11 co-occurring lineages were detected during this period, as shown in the bottom left, which were used to generate a total of 42 time-ordered mixtures. Also, as shown in the bottom right, 12 co-occurring Omicron and Delta sublineages and their parent lineages were detected, which were used to generate 36 time-ordered mixtures. Importantly, these mixtures contained many low-frequency values, while the mixtures generated from the NextTrain data additionally contained closely related lineages and sublineages. Both these aspects are important for stringent testing of the computational methods. Next, starting from the obtained virtual mixtures, one genome per lineage was picked and reads were assimilated from these using two instruments, ART and SWAMPy. ART uses empirical read quality profiles summarized from large real sequencing data. The values used in the simulation are listed on the left. One art parameter at a time will be altered to simulate a total of 236 samples with different read length, different total read count per sample, different Illumina platforms corresponding to different sequencing error rates, as well as error-free reads. Subsequently, we will evaluate how the performance of the benchmark methods is affected by these parameters. To note that the preliminary benchmarking results presented below are based only on error-free reads. To simulate wastewater-related properties, we use the SWAMPy simulator. As illustrated, SWAMPy starts with genome sequences and their corresponding abundances, then creates an initial amplicon population and generates the corresponding number of copies per amplicon, simulating variable amplicon abundance, including amplicon dropout, as controlled by a user-defined parameter that reflects the quality of the wastewater sequencing data. Next, SWAMPy mutates the sequences to introduce errors related to RNA degradation in wastewater or induced by PCR or library preparation artifacts. In the final stage, it uses ART to simulate reads along with sequencing errors and base quality scores. A key advantage of our benchmark will be assessing method performance across a few real datasets sequenced with 3 different technologies, Illumina, Oxford Nanopore, and Ion Torrent, as well as using different amplicon panels. The datasets already available are marked with a check mark, and the number of mixtures planned for each condition is indicated in the brackets. Importantly, mixtures with predefined composition will be sequenced directly, but also spiked into raw wastewater or RNA extracted from wastewater to separately assess the effect of the wastewater matrix, but also of the wastewater genomic background on the quantification accuracy of the methods. Just like the simulated data, these mixtures will also contain low abundance lineages and closely related ones. In addition, we will prepare lineage mixtures from inactivated SARS-CoV-2 viral particles, which will be sequenced directly but also spiked in wastewater and sequenced. We expect to have about 200 real sequenced samples in total. Our preliminary results were obtained using the dataset with no sequencing errors and a small subset of listed methods, the repurposed RNA-seq method Salmon, RSAM, Callisto, which uses expectation maximization, and the deconvolution methods ALCOV and PGX, designed specifically for wastewater surveillance and which use the least squares method. This plot shows the absolute error or L1 error for each method, defined as the sum of the absolute values of the differences in percentages between the true abundance values and the predicted values. It can be noted immediately that for lineages, shown to the left, the absolute error is much smaller than for sublineages and their parent lineages, shown to the right, despite the fact that it is summarized over a larger number of mixtures. This is valid for all methods listed. In addition, RNA-seq tools RSAM, Callisto, and Salmon show better accuracy than the convolution tools ALCOV and PGX. To note that all methods listed here, except for ALCOV, use the same user-sourced references, allowing a proper direct comparison between these methods. By contrast, ALCOV only allows to use its own reference database, which has to be taken into account when comparing it with our methods shown here. The distribution of absolute errors confirms the same pattern. In this plot, where the horizontal axis indicates the absolute error threshold, while the vertical axis the corresponding percentage of values below this error threshold, a steep upward curve indicates higher accuracy. While for lineages, most methods show good or very good performance, for the mixture of sublineages and their parent lineages, they perform much worse. In all cases, RSAM and Callisto are the most accurate methods, while PGX the least accurate. Plotting the true frequencies against the estimated frequencies show that the lineage quantification is fairly accurate across the methods. For sublineages and their parent lineages, however, the methods show lower accuracy, with RNA-seq methods based on expectation maximization, bottom row, performing better than the convolution methods based on the least squares, upper row. So in conclusion, repurposed RNA-seq tools RSAM, Callisto, and Salmon seem to yield more accurate results than the specialized wastewater surveillance methods tested here, ALCOV and PGX. Quantification of closely related sublineages and lineages seems a greater challenge for the assessed methods. Further, we plan to evaluate a number of metrics for lineage calling and lineage relative abundance estimation, which will be contrasted across the broader list of computational methods, but also across the different datasets described earlier. In addition, we will assess the computational performance and user-friendliness of the methods. With this, I would like to thank all the people collaborating on this project, in particular those who guided our efforts, who helped generate the datasets and to run the computational methods, but also those who helped with the project otherwise.